if you're all ready and set to go.
We will start in about another minute or so.
Okay.
Give people a chance to get on. Also, this is recorded because we got people all over the place, and a lot of people watch these things asynchronously. So at better times for them, and so yeah, I'll just let it go for seconds here, and then we'll get rolling. Message here. Okay. So I should ask you, Marshall. Now, would your plans be to stay in the Purdue area, or would you be wanting to move to Lakewood, or our Colorado office or Tucson office down the road after your postdoc is done?
Yes. Yes. I'll consider the possibility of moving to Tucson or to Colorado. Is just fine. Yes.
Yeah. Yeah. They're both nice places.
Mm-hmm.
Now, I sound funny this morning because this side of my face is numb. I had a crown put in, so.
Oh, yes. Okay. You had dental surgery.
Well,
sorry about that.
yeah.
We'll get there.
Or just dental abuse, I think, I'd characterize it. So, okay, let's go ahead and get started. I'd like to welcome Dr. Marshall Tabetah. He is postdocing at Purdue and has applied to join us as an associate research scientist. And his materials are posted on our internal site. And very much looking forward to your talk. And Marshall, why don't you start off by just giving some of your own background, and then proceed ahead. So I'll turn it over to you.
Yes, sure. Yes, the topic I'm presenting today is I'm presenting work on multi-analytical approaches that would enhance and predict survivability as we plan to be in space, over longer duration missions. I'm talking a little bit about myself. Yes, my PhD is in electrical engineering from University of Virginia, earned in 2014. And there I did a lot of molecular interaction modeling for nanotechnology applications. And so I did develop models and simulate them, and did come up with some important breakthroughs in mechanisms that led to formation of nanoparticles for nanotechnology applications. Then later on, I did a postdoc in Purdue where I did some computational modeling of meteorite fragmentation in Earth's atmosphere. And we came up with a mechanism that explained how the Chelyabinsk meteorite eventually got fragmented upon entering the atmosphere. And then later on, now I'm doing a second postdoc currently, where which makes the topic of my discussion today, which engages multi-analytical approaches to enhance survivability in space missions. And my current advisor is Dr. Porterfield, and for the first postdoc, it was Henry Marsh, who unfortunately has passed away. And so, yes, a lot of my work revolves around modeling, programming, developing algorithms, even for electronic circuits especially. So yes, I have background in multiprocessor computing and parallelism, speeding up high performance computing using things like OpenMP or MPI, as well as using shell scripting, for when I want to organize, and submit several jobs at the same time, and as well as organize them. And definitely in the electronics area, I do program microcontrollers, do program Arduino microcontrollers, PIC microcontrollers. And very interesting, yes, I do program FPGAs, which is something not quite easy. Although FPGAs offer a lot of advantage in speeding, creative speed than microcontrollers, because you can do concurrent coding. And there I use VHDL language. Of course, I do data processing as well, most recently using R and Python. You use these a lot to process things like biological data, things like omics data. You use a lot of R and Python. And then, yes, there is usual scientific programming, which you have to do when you're relating to modeling. And so Fortran, I've programmed a lot in Fortran as well as C and C++. And then you have MATLAB, definitely must use that for some analysis in science programming, and Java. Yes, I have. What have been my research interest in the past few years? Yes, a lot of FPGA design and microcontroller instrumentation. I've been quite interested in remote sensing. This is quite interesting. How do you integrate components in a smart way that can facilitate and enable remote sensing, data acquisition and remote sensing, even whether it is in space, right? Especially when it comes to space, or even if it's just on Earth, like being able to sense a remote agricultural field or a remote event, in a smart way. And yes, continuing my research interest, particle interactions modeling and simulations, what I've been doing, for many years, be it with molecules or at a level of fluid mechanics, which is a bigger length scales. Yes, I've been interested in recent years in radiation measurement and countermeasures. When we go to space, it's important to stop, the intensity of radiation from doing damage. And recently, with the Nuclear Electronics Lab at the International Atomic Energy Agency, I've had some collaboration where we try to resolve radiation particles that closely overlap within microsecond resolution. And yes, I do have some interest in machine learning and artificial intelligence in some of my current research projects. So pretty much that represents what my academic and research background is. Now, let me move to
Marshall, are you using artificial intelligence in some of your work these days?
Yes, we're planning to use artificial intelligence algorithms in a number of projects. When we have omics data is what? Biological molecular data. When we have such data and we have them from different organisms or from different experiments, we want to see how we can correlate the data to derive more useful meanings. And in such cases, you have to use artificial intelligence data. Things like machine learning, and you can also use machine learning to classify data. And this is going to be helpful in something like disease prognosis, whereby you can say, if this data has been behaving this way, against the set of data that has been behaving this way, there's a trend to say that we can classify a clinical outcome against one another. Yes. Okay. Yeah. So, so the challenges that we're mostly trying to do currently with NASA and many other space agencies. The eventual goal is to, in the future, to be able to thrive and survive in space, in the Moon, in the Mars, or in other asteroids that will be further explored. Okay, and so putting up a space habitat is something quite important. Question is, how would astronauts, how can life survive long-term in space? With this, what are the main challenges? You can talk if, space is a hostile environment, right? So we do not have, for instance, the same, we breathe here, we have an atmosphere which has a given pressure, and we can breathe oxygen and release CO2, but space does not have all that environment. It does not really have such an atmosphere, especially even when you go to the Moon or to the Mars, where you have a highly reduced gravity, partial gravity. We do not have that. Although one may say that we can still cope with such environments because we can supply our own air, we can supply artificial air to rely on. But however, what are two main challenges remain, that still have to be overcome if we want to have a sustainable life in space. And this is microgravity, the fact that when you're in the ISS, there's no gravity at all. Or as I said, in the Moon, when there is partial gravity, things do change. The physics does change. As you can see, an astronaut is weightless. And so the physics in the body, the physics to the body, the human body becomes different. We're going to see this later on. And also radiation is a big problem Yes, and so in later slides, we're going to see how these two effects will impact human life and human health. Yes, when it comes to gravity, the first thing we have to remind ourselves is the behavior of the candle. When we light up a candle in a normal gravity environment, we can see that conic, that conic shape of the candle as it lights up. But if you switch off gravity altogether, that candle adopts a spherical shape. And this is all because of the process of combustion. Gravity drives buoyancy. Lighter gases tend to move upwards while the heavier ones are downward. So when this candle heats up its surrounding air, the surrounding air will drift upwards. As it drifts upward, that drives the shape, that drives the somewhat conical shape of the candle. And so if you take off this gravity altogether, you're going to have a situation in which the surrounding air is hotter, but it's no longer light, it's no longer buoyant, that it's not driven upwards. And so all what it does is it would expand in all directions. And so the candle will retain a spherical shape. And so with this, we understand this is just to enable us to understand, gravity-driven buoyancy. And so when you get out there in the International Space Station where you have no gravity, you have a similar effect. Gases can no longer be above liquids, and so they tend to mix up with the liquids. And what happens? You have what we call slug flow. So gases can now, coalesce with one another. They will also they will block, liquid flow as they coalesce with one another. And so you have things like liquid holdups in capillaries. And this is a completely different effect from what you have on Earth. And so this is a challenge now. Our own bodies are made of capillaries. And then, so these effects happen in our body. We have fluid shifts and all of these things that we have to face out there in space. And even in the systems that we control, these phenomena prevail. And so there is definitely the later story of an astronaut, Luca Parmatini, who actually he got drowned in his own helmet when he tried to make a spacewalk, around 2013. And this is because there was something, one of the water filters in his spacesuit got damaged. As the water filter got damaged, water now could easily flow all around his body, because when we're on gravity, the water will generally gravitate downwards. But out there in his spacesuit, he's there weightless. The water could easily flow around the body, and then it's got into his helmet. It got into the head, it got into the region that surrounds the head. And so it kind of, that water filled up his helmet, and then he struggled, he could no more hear. It clogged his ears, and he ended up struggling, having to breathe. So he almost got drowned if he was not really a careful person. That he got blind even, and he almost got drowned, and he could even die as he struggled to make his way, back into the space cabin. So these are effects that tend to happen. These are challenges that tend to happen when gravity is altogether taken away. Okay, so fluids and gases just really do some more crazy stuff. And then with radiation, what happens? Yes, when we go to the ISS, we still shielded from cosmic radiation by the Van Allen belts. The Van Allen belts are the regular magnetic field that we have around the Earth. This would shield, any cosmic rays from impeding on us when we go out there in the low Earth, insofar when we go as far as the low Earth orbit. And then the space capsules are also designed in such a way that they can stop any impeding radiation. So somewhat we are shielded effectively from radiation, but still measurements and analyses upon life, be it mice or astronauts, analysis have been done to notice that there's still been an effect and there's been an effect of radiation on some of our organs, especially the brain. There have been cognitive issues with the brain. And so somehow radiation still makes way into the space habitats out there in the ISS, onto the space cabin. And of course, what are the reasons? Solar particle events, are really difficult. They're unpredictable. Solar flares are unpredictable. And those solar particle events can easily make their way past, any shields, be it the Van Allen belts or be it our capsules. And also you have those high ionizing particles, like the neutron rays, like the neutron particles or proton particles. They can still make their way past, the space capsule to the shield. And so this is something that is worth concern. And so being out there in space, we can easily accumulate radiation doses that highly supersede what we can face on Earth. And yes, and when we get into outer space, the moon and the Mars, we lose protection. We've gone beyond low Earth orbit and we lose protection from the Van Allen belts, and it becomes more damaging. Yes, we become more exposed to galactic cosmic radiations. This is just nuclei. These are nuclei that are traveling at speeds that are close to, the speed of light. And this is really high. This is really ionizing damage from such particles is very high. And so these are things that we have to be, we have to account for in terms of our health and survivability. Yeah, so when it comes to the work that I've been doing, and I'm part of the Genelab analysis working group that was created by NASA to process and analyze space omics data from the repository that Genelab itself is hosting. And of course, the goal here is to determine the impact of spaceflight on health. So here we try to establish, phenotypic changes. We don't want to probe, astronauts' health just by detecting the symptoms. That's a phenotype, that's a general phenotypic approach, which is usually adopted in many hospitals. We want to be able to, since in space we don't have, the medics are not there, to all the time determine those phenotypic symptoms. All of this availability is not there. We want to be able to extract blood, from the astronauts, and especially from mice, which are more affordable, extract blood, and then store the samples, freeze them when they get here on Earth, collect data from them, collect genetic data, genomics data, biomolecular data, and then we try to establish from the analysis of those data, we try to establish the phenotypic effects. We try to establish what our life specimens have been going through. If you have any questions, please. Okay. All right, let me move on. Yes, and so, in one of the projects in which we have been working in a research group, we tried to determine the impact of spaceflight on insulin. So we know that gravity is no longer there in the ISS to exert a force on our muscles. And so our muscles may not build up as they build up in gravity. And so there is an interesting question. What is the role of insulin and estrogen in maintaining muscle mass, given that there is no microgravity? And so in this project, we took, omics data from muscles, both rodents, that is both mice and human muscles. We took we took data from many spaceflight data sets, omics data, and we tried to determine what are the clinical outcomes, what were the clinical outcomes, from these data sets. And so we tried to determine pathways, insulin and estrogen pathways that were impacted. So again, what is insulin? Insulin is what regulates, the glucose level in blood, right? You need glucose uptake in cells, which is then oxidized, right, to produce what? ATP that is stored as energy. And energy is what we need. So if we don't have any energy, our muscles cannot do any work. And so, yes, we did all of this analysis. We tried to determine how differential expression of genes, in spaceflight compared to grounds, how the differential expression was altered. And so there is definitely, we did show some differential expression. We did show that when you go out there in space, many of these activities, many of the aspects of insulin signaling and estrogen signaling are being altered in space. And so there is definitely some further research that can be done in this area. And yes, we came up with a review article out of all this work that did show all of these results. And this article has been published in Nature Communications Biology. Well, it's not yet published, it's in press, that it's been formally accepted, and it will be published in Nature Communications Biology. And that is the list of authors. And in another project, as I was talking about muscles, it's very interesting how spaceflight affects frailty. Here, we define frailty a little bit different from aging. You know, we age when we stay much longer on Earth, because as we stay much longer, some of our physiological reserves become depleted. But frailty is that state of vulnerability, is that clinical state that defines, you know, how much of our physiological reserves have been depleted, how much we can bear stress. So when we go to space, how much of these effects do we notice? So again, we try to determine the phenotypic outcomes using omics data, using transcriptomics data, with this entire team of people that come from so many different universities. And yes, we did try to look into the underlying pathways, you know, in the body that relate to DNA, to mitochondria, to chromosomes, to see how frailty is impacted in spaceflight. And we did come up with very interesting results. We did notice how many of the biomarkers, when I talk about biomarkers here, I'm talking about the genes that I usually upregulated in frailty. And so many of these genes were regulated. We did use a network of methods, right, in order to come up with these analyses, make and produce results, right? So from the data sets, we used data sets and used several methods like gene set enrichment analysis. And we did come up with very important results that predicted several frailty outcomes. And although there are many things I will do for this, such as prediction and classification, in order to see what is a general frailty index when they come out. That is to say, out of all these results, how can we determine a frailty index for those who go out there in space? And we did come out with very interesting results, even definitely to say, yes, there should be some further investigations, not just in frailty, but it's also in sarcopenia. Sarcopenia mostly relates to muscle mass as opposed to frailty that is generally determining the overall outcomes in terms of our vulnerability to stress and all the space-related issues. Okay, so, and in all of this, we did put up results out there, not yet published, but we did get some work done. And so this is a published preprint, which just means that it will be published after the journal finishes the review. So this is a preprint from Nature Scientific Reports, in which we did show that there is some impact of spaceflight on frailty biomarkers. If you have any questions, I'll be happy to answer. And so yes, Andrea Camera, who has been leading this project, did present some results in the ESGS IRO conference in Houston. Okay, so those are two projects in which we've tried to see how spaceflight impacts health conditions, based on transcriptomics data. And of course, one other phase of my research has been involved with plants, because we need plants to improve, we need plants to improve our life out there. in space, be it in the International Space Cabin when we may not have a lot of supplies, coming from Earth, or be it in the Moon or Mars where we just need to establish a habitat that will depend on plants for life. So not only do we depend on plants for food, we can also depend on plants for our respiration, because plants produce oxygen as they take in our exhaled CO2 in photosynthesis. Okay. And there is also another way of looking at things, that is just the waste that we produce from our bodies can be input, it can be fed into a bioreactor that would use this waste and convert them into nutrients that would be used by plants. So that's another cycle in which things can be looked upon. And so there are experiments to see how all of these, how such systems can be put in place. So of key interest to us is what are the primary or say secondary effects of microgravity? How would gravity affect the way plants grow and reproduce? We know that some of the primary effects, gravitropism, how do plants, as I said, sense gravity, or how does gravity affect the flow of fluids in plants, and how does that fluid distribution affect the growth of plants? And then you have secondary effects, which is how does that fluid distribution relate to the biophysics at the molecular level, and how would it affect the physiology in plants? So it would be of interest, it would be of interest to know how the lack of gravity, or to say how the lack of gravity will affect many of those physiology processes of interest in plants, such as transpiration or water loss, the photosynthetic gas exchange between oxygen and carbon dioxide, heat dissipation, mineral nutrient uptake is quite important. When you get to the roots, there is a rhizosphere root exudation. So when we are in microgravity, where buoyancy-driven convection, or let's say gravity-driven buoyancy is absent, how do all these processes in the physiology of plants, how do they occur? Yes, and so it's quite interesting, because now we know that the oxygen, or it may be CO2 in the night, but during the day is regularly oxygen. The oxygen that leaves the leaves of the plants will generally, if it's in a gravity environment, if it's in a regular gravity environment, it will leave the environment of the plants through buoyancy, through the flow of buoyancy. But when we lack now gravity, the exhaled gas will stick around. And so the exhaled gas will stick around the leaves, and so they may form a boundary layer. And so it will be interesting for us to determine the thickness of this boundary layer. And so when we have a layer of gas around our leaves, how will that affect, the respiration of the plants through the leaves? This is an important and interesting question. Yes, and so in relation to plant growth, yes, we've taken some measures. We're interested so when we have plants out there in outer space, being in the ISS on low gravity environments, we want to be able to track the motion of these plant parts. When we track the motion of these plant parts, we can then somehow characterize the behavior of the plants during growth. And when we characterize the behavior, we may be able to tell, how the plants have responded to the environmental conditions. And so we use an algorithm, to track the plant growth. We use an algorithm, a deep learning, this is already now in the area of artificial intelligence. So we use now a deep learning framework, to track, that has been typically used to track, the motion of small animal parts. but here we're adapting it to track, the motion of plants, because the resolution could be good enough. Okay. So the way I implement sleep, I just do it. Yes, I do it with Python code. I use a deep learning framework, and then I do for some instruction purposes. I do some labeling. Whenever you're doing a deep learning or machine learning algorithm, you have to label something, you have to label some items, and then you submit those items into machine learning framework. And then these labeled parts will train the algorithm so that it will then identify all the parts that have not been previously labeled. And after you have labeled everything, the next thing you do is, you label finally by prediction. Then the next thing you can do is you can now retrieve the coordinates, right, of the different paths that you have labeled in time, right? When you feed a video now, you can then predict now the motion of all the parts in time. Okay, and then you can definitely conduct scenarios to improve the algorithm simply by relabeling what has been predicted already. Okay, if you have any questions, please. Okay. So I may not enter here into any details, but if you're just interested into any particular details about the algorithm, they are laid out in this slide. We use things like a cloud server, because to run a deep learning framework, you may require a lot of computer resources, and so we just put that in something they call Google Colab. Google Colab is a free cloud-based server with all of the heavy resources that are needed to train the algorithm and predict the results. So in an example scenario here, these are plants that are grown in space. A specimen of them is grown in space, and another sample is grown on ground. So this is what you call Arabidopsis, the Theliana, and we take a video of such plants, and then we try to contrast. We're going to contrast the difference. If we grow them in space, how is the growth pattern different from the growth pattern on ground? And so all what we'll do is we'll submit such video into this algorithm, and then we'll try to track the motion of this algorithm, and then we'll try to establish the differences.
And how long are you tracking them in space?
Okay, when they're out there in space, good question. So since they are out there in space, you just turn on your cameras, and then you record a video of the plant. And then when you're recording the video, you have to record the time frame, right? The time lapse between your images. And so you just record them, and then you'll send them. You can relay them directly to space, or you record the data, and then later on we'll do the analysis on ground.
But are we talking about weeks of time, or months of time, or a year of time?
Yes, very good question. This could be several days of time, and so it could be weeks, because what is happening here is you have to make sure the entire plant grows. You have to track it from the time it's growing, right? Mm-hmm. From the time to the time it fully blossoms. And so it could be weeks of time. And of course, depending on the type of pattern that you are interested in, right? That would determine the duration of your tracking, right? I may just be interested to see how the stem or the shoot is wavering, in some phase of growth. Then I may just be interested in that duration. That's right. Okay, so in that case, what I did is I did something a little bit primitive out of this, and then I exported the data from Sleep to, let's say, to MATLAB or to Python, and then I tried to, all of that is in a matrix, and then and then the next thing I did is I could now plot because I have tracked the coordinates of the different parts in time. So in something very primitive, I tracked the stem, the taproot, and the base stem. And then what I have plotted here is the y coordinate. So that's the y coordinate of the stem against the taproot and the base stem. So and then that is the x coordinates. Again. And so this is just a primitive way to show that we can track the plant parts, in time, and we know the duration, we know the time lapse between each frame, right? We can track the motion of the plant parts,
So the x-axis is a distance and the y-axis is something else?
So yeah, so this is the y. Yeah, so something, yes, that's right. So something we need to know is we need to know the time lapse between the frames so that we can know the time between, yes, and we need to also know like you just mentioned, the distance, right? What is the separation? What is the spatial separation between pixels? So what is the spatial separation between coordinates? Because here I have put the y coordinates, the y coordinates, that's on the y-axis, right? But here, I'm just telling you that's the pixel. Yes, but I've not told you, for instance, how much each pixel represents, what the spacing between each pixel represents. That's right. Yeah. Okay. Good. So after the possibility, so after we have tracked the coordinates, in space and time, right? We can then do things like start, deriving useful patterns out of the growth. We can do things like we can run machine learning algorithms like K-means clustering. You can run them on the velocities and the clustering methods to determine, important growth patterns in the plants. So we will engage more machine learning or deep learning algorithms to study the growth pattern in plants. Yeah. And of course, in another phase of our work, yes, which relates to human respiration, and that really closely involves microgravity. We know that while we're here on Earth, right, we have a lot of, as we breathe in oxygen, we breathe out CO2. We have a lot of natural ventilation, especially when we stand outside, we have a lot of natural ventilation. And again, gravity-driven buoyancy gravity-driven buoyancy that keeps a lot of air circulation is quite important, as you can see. The wind, how the winds blow, the winds blow mostly because they are lighter, lighter air particles, always rise up. And so all of this motion, this natural motion helps a lot in maintaining some natural circulation. So we always get some natural fresh air. Yeah, we always get some natural fresh air in the natural environment. But if we go out there to microgravity, we lose this gravity-driven buoyancy. And how do things change? So coming back again to the candle, as we explained it from first principles, this is something that we have to account for. And when we go out there in the microgravity environment, so the lighter gases will no more be able to rise. We'll just have things can transport when mass transport cannot be driven by energy, by kinetic energy, or driven by momentum, it becomes only driven by diffusion, right? It becomes only driven by diffusion when buoyancy has no role to play. So out there in space, yes, in microgravity, you notice that if you breathe out CO2 and there is no way for it to be driven out, and why can it be driven out in regular gravity? What we breathe out is hotter because we are 37 degrees inside us, right? And outside we have about 25 degrees. So when we breathe out, it's hotter. And so this hotter air is, well, because it's hotter, And so less denser, it's lighter than the air outside. And so it will generally be buoyed upwards, giving room for us to take in fresh air. But if we're in a microgravity environment, this air will just expand, it will just expand outside of us, and it can form a bubble, around us. And as it forms a bubble around us, it can stress us, okay? So, yeah, that is it. And so that is what happens. So, as I was saying, if you're in gravity on the left side, you see the air that you breathe out, it rises. And so also remember that because we're 37 degrees inside and 25 degrees outside, the air that is right on the surface, on the surface of our body is hotter, and so it also rises upward. This is what creates what they call our thermal body plume. So our thermal body plume has a convective role that also facilitates, to clear away any air or any pollutant particles that are close to our nose or our face during the breathing process. And so you can see how any air that is breathed out, the CO2 that we breathe out, in a simulation, in a simple 2D simulation, the CO2 that we breathe out in the case of gravity rises upward. That is it. So it rises and remains out there. But when you're in microgravity, which is on the left-hand side, on the right, there is no buoyancy. And so when the air that we breathe out loses its initial momentum, it can only diffuse slowly outward. And so we can have accumulation in successive cycles of breathing. And so this is something that is happening in the International Space Cabin. There have been reports of headaches in the International Space Station, and all of these have been associated to high CO2. They have measured CO2 levels to notice that 0.5% CO2 levels linger in the cabin, despite, measures to scrub off the CO2. There have been chemical procedures, CO2 would easily react with lithium hydroxide, yes, would easily react with lithium hydroxide in an exothermic reaction. And so this is what they use at the walls of the ISS cabin to scrub off CO2, but still it's not enough because a lot of CO2 accumulates in the space cabin from exhalation. And so this is a challenge. This is a challenge that has to be demonstrated. So one of our coordinating interests is, first of all, want to show it to the community, make it clear that how microgravity enhances the accumulation of CO2 in microgravity and in the space environment. When people become more aware, then can take on challenges, to find ways to reduce that effect. Yeah.
So don't they use fans?
Yes, fans. That's right. Yeah. So yes, exactly. So the immediate idea that comes to mind is fans, and this has a history. Previously, fans were used, and at some stage of time, they stopped using them. But another important question, when we run the simulations and it is now more evident, how the CO2 bubbles form around the head, it becomes now more obvious where you have to apply a fan. You have to apply a fan closer to the head, right? Good. You have to apply a fan. So if earlier you applied fans, in random places, that fan will only circulate, will only move the air around. On average, you still have the same amount of air that moves around. You may have to apply fans closer to the head so that fan will force CO2 to move away from the head. When it forces CO2 to move away from the head, you create more pressure upwards, right? So that pressure upwards, according to Fick's second law, will easily force the CO2, closer to the walls where we can, and then when we get to the walls, we can activate the removal of CO2. That's right. We can put up an idea like that. Or we can put up mechanisms that buoy up, yes, CO2 from the environment. That's right. And so, concluding this talk, we have to think of, the milestones in human space history. We all started, very primitively when we had to launch, when we had to go to the moon. In those days, the health effects were not quite important, but with time, especially when we got close to 2000, it became more obvious that health is an important issue, as many of these things were monitored. Health became an issue. But most importantly is that the technologies that have been born out of, space research, and the technologies have led to many commercial spin-offs, have led to many innovations that have been implemented in our industry. So basically, our industrial innovation has, in recent decades, has partly depended on concepts that have been innovated in space. And so this is very important to the commercial industry, besides being important to survival in space flight. And of course, yes, it is important to be able to create a stable commercial economy and try to avoid any gaps in any current gaps that exist, such as delays or stagnations in trying to develop any needed facilities in the International Space Station laboratories. So, so that will be, so this basically concludes our talk. If you have any questions, yes, I'll be happy to answer them. Thank you very much for listening.
Thank you, Marshall. Really interesting. Any questions, Marshall? I have a number of questions, but one is interesting seeing about the plant development. I recall some Russian experiments back in the late 80s that were a little primitive, but they indicated that nerve muscle junction development in fetuses did not work well in mammalian life in less than 1 gravity. Has there been any work that you're aware of about like, rat development in 0G or in low G? I know they haven't had the centrifuges really needed to do that, but I was wondering if you knew if anything has been done.
Well, just a tiny bit. Will you?
The effects of low gravity on fetal development in mammals.
In mouse, development of what again?
Yeah, fetal development in mouse. The effect of or any other small mammal. The effect of low gravity or 0 gravity on fetal development. There was some old research from decades ago that suggested that fetal development did not work well in less than 1 gravity, which of course has a lot of implications about our future as a species beyond Earth.
Let me just do one thing. Let me just do one thing so that we can. Let me just do one thing a little bit. Let me augment the volume here. Yeah, like you said, let me just augment the volume so that I can capture you quite well. So you said, yeah, the Russians, they did an experiment to determine the effect of gravity on the development of some organs in mouse.
Yeah.
Okay. What organ was this?
Well, it was nerve muscle junctions.
Nerve muscle junctions. Okay. Oh, okay. So good. Interesting when it comes to, yes. Okay.
Maybe there hasn't been any research on that since then.
Okay. Oh, nerve muscle junction.
Yeah, probably not.
Yeah, well, although we did investigations on mouse muscle, yes, and
that would be for adult mice, right? Not for developing mice, to see how it affects the development of of muscles from from fertilization until birth, and then development from birth to adulthood.
Okay. Because yes, when it comes to the nerve, we have to relate things to cognition, right?
Well, not in
Not in mind.
mine. Yeah, no. I mean the focus on the effects of space on species and humans and stuff has all been adult. There's been, to my knowledge, I'm not aware of much, if any, but I haven't been paying that close attention to it. Work on the effects of space, particularly gravity, on development of animal life. And
Yes.
But if we're going to talk about doing something more than just going to the moon and planting a flag to establish population of people beyond the Earth, this is kind of an important area that somebody at some point is going to need to decide that we need to figure it out.
Yes. I do think but really related to the work we do, this comes a little bit again to the omics work that we do, by studying by revealing how genes are expressed in those conditions. We're able to predict how the behavior of those genes will evolve into outcomes, like you were saying, into developmental outcomes of the mice.
Yeah,
that is right. So yes,
well,
through this transcriptomic study, yes, at least
Oh.
That's how close we got.
Yeah. No, yeah. Well, I imagine that your work is also important for understanding just to what extent people, were the possible effects of spending how much time out, like on the surface of the moon. If you're up there for a long time, then there's other factors that need to be taken into account in determining the safety of exposure to this safe environment.
Right. Yes. Because yes, when astronauts they return, there is definitely some recovery from some of the spaceflight effects, because those effects have been witnessed on the health that they do recover in time. But there are long-term effects that are not erased. Those that are built in the DNA. DNA damages, mitochondria, like subcellular changes are very hard to reverse.
Yeah, I think DNA damage is just scary.
Yes.
Anybody else have any questions for Marshall? Okay, well, hey, thank you very much for your talk.
Good.
And we'll be in touch.
Okay, thank you very much for listening to all this. Okay.
Yeah, very interesting.
Okay, thanks. Goodbye.